Thanks, Alex. Yeah, Brian asked me how many genes I'm going to talk about, and my answer is one. I'm only talking about one gene, but he's going to talk about more. And Alex, yeah, I think this won't be the hardest gene we ever see, but it is difficult. So I'm Scott Hickey. I'm Senior Director on the Commercial Applications team. And today I'm going to talk about some work we're doing in a recent project looking at this really complicated array called D4Z4, and it is in a type of muscular dystrophy called FSHD. So these are my disclaimers. And first, I'm going to do a little bit of background on FSHD and what it is. So it's facioscapulohumeral muscular dystrophy. It's genetic muscular dystrophy. It's caused, or it's characterized by progressive skeletal muscle weakness. And it's the third most common muscular dystrophy, affecting about 1 in 8,000 individuals. The severity of symptoms and also the onset of symptoms can vary, but the genetics are relatively well understood thanks to decades of research into this disease. So the pathogenic gene in this case is called DUX4, and it's located on chromosome 4 right at the end, right at the telomeric end, in a really challenging region of the genome, and also in this repetitive microsatellite region called D4Z4. It has two types. So FSHD1 is caused by the repeat contraction in this region, and that's about 95% of cases. FSHD2 is similar, has the same clinical phenotype, but is a different regulatory pattern and a different genetic mechanism caused by variants in epigenetic regulatory genes and methyltransferases. So this is what the microsatellite array looks like in a healthy individual. This is anywhere from 10 to 100 repeat units, and the repeat units are 3.3 kb, 3.3 thousand bases each. So in a healthy individual, this can be up to 300 kilobases long. And it's made up of these D4Z4 repeat units that have been coupled to a partial DUX4 gene. And then there's a DUX4, full-length DUX4 cytotoxic gene at the end of the array. In FSHD1 affected samples, the array is contracted or deleted, and it contains 10 repeat units or less than 10 repeat units total. The mechanism of action here is DUX4 is expressed. DUX4 is a cytotoxic gene, cytotoxic to muscle tissue, and it's expressed in FSHD1 because of this repeat contraction. The long form of the allele is methylated or hypermethylated, and at each one of the D4Z4 repeats, in FSHD1, the contraction clears the methylation and it leads to hypomethylation. So then DUX4 is aberrantly expressed in muscle tissue. To complicate this further, so D4Z4 appears all throughout the genome, but it also has a very similar, highly homologous region in chromosome 10, 98 to 100% identical. And the 10q allele also has this D4Z4 repeats. So you really have to know which chromosome you're looking at here, and they're very, very similar. Also, the repeat units between each other are almost identical. So this is a really difficult region to sequence and analyze. Individuals can have all possible haplotypes, and you only care about the 4qa allele because that's the one that has a functional copy of DUX4. The reason for that is the 4qb and the 10q alleles all have a pathogenic mutation, a non-functional mutation in the polyadenylation region. So those don't form stable transcripts. So in order to diagnose FSHD, you have to know that you're looking at the 4qa allele only, and the other alleles don't form DUX4, don't express DUX4. FSHD2 is slightly more complicated, a slightly different mechanism. It also expresses DUX4, but it doesn't depend on the repeat contraction. It doesn't depend on the length of the repeat. What happens in FSHD2 is you have a pathogenic variant in another epigenetic regulator or a methyltransferase. The most common one is SMCHD1, and it knocks out methylation across the entire locus. So DUX4 is expressed, but you can have the long form of the allele, so you really need to know the methylation status of that 4QA allele in FSHD2. Okay, so how can we characterize these samples with Oxford Nanopore long reads? FSHD testing traditionally requires multiple simultaneous assays, and that's because you have to size the repeat expansion and contraction. You have to call the variance for the for FSHD2, and then you also have to look at the methylation signal. So traditionally, this is optical genome mapping, southern blotting, and short read based testing. With Nanopore, you can do all of these things. You can get the 4Q haplotype, you can get the D4Z4 repeat length, and you can also get the methylation and variant calls all from a single test. If you're not familiar with nanopore sequencing, this is how it works. We take a strand of DNA and thread it through our nanopore. It's embedded in the membrane, and the electrical signal that is caused, we convert that into our base calls. The approach is PCR-free, so we have native strands of DNA, and that means that we can get the methylation signal directly and base call that. That comes for free with your base calling. And also the reads can be of any length, so you can have really short reads to medium reads to long and also ultra long reads, which I'll talk about in a minute. For this project, we took two sequencing approaches because Nanopore is very flexible in terms of the target size of reads that you want. We can take two approaches for this. We can do our standard kind of workhorse chemistry, which is called ligation sequencing. That gives you about 30 kb reads on average, and is really just our normal standard whole genome workflow. But you can also do a prep called the ultra long prep, which gives you 100 kb plus reads up to megabase reads. Both of those protocols are available on our website. The ultra long requires a little bit more finesse on the lab side, but you can get really, really long reads with that workflow. So we decided to do both for this project because we wanted really long reads that span the entire D4Z4 microsatellite arrays in all of the samples, but we also used the ligation sequencing chemistry. So we took some FSHD cell lines from Coriell, we extracted the genomic DNA, and then we can prepare our sequencing libraries. Again, all of this is on the website, and then we can go and do our downstream data analysis. This is just a summary of what the sequencing data looks like. So on average from our LSK sequencing, that's in blue, that gives us about 30 to 40x coverage per flow cell, and we get read lengths in the 30 to 40 kilobase range. So that's average read length. And then in the ULK is shown in purple, we get about 20 to 30x coverage, so not quite as high, but the reads are a lot longer. Those are 100 kb plus, up to 122 kb for one sample on average read lengths. So then it's time to go in and do the counting of the D4Z4 repeats. So how did we do that? We took two approaches. We have the ultra long reads. We can basically take those directly into a software called D4Z4 end-to-end. So that was published previously this year by a group in Australia. And that we can take the ultra long reads directly into that, and it counts the repeats after aligning. So we use that as our truth set because these reads are single reads that span the entire array and can be counted. So we think that those are really, really accurate readouts of the D4Z4 locus. We also take our LSK reads, the ligation reads that are slightly shorter, 30 kb, and we can assemble those first. So we use the Hi-Fi SM assembler to assemble contigs out of those reads first, and then we put those into the repeat counting software, and it works the same way. Then we can compare the results to each other and do downstream methylation analysis. This is what it looks like for a representative sample. So this is an FSHD1 affected sample with a 4QA allele, and it has the contraction only in one allele. So it's 4QA, but one of them has 7 repeat units and the other one has, I think, 35. And so we can haplotype these correctly and count the repeats correctly in both of our workflows. We get this from our LSK assembled data, and it matched to our ultra long read data. Importantly, we can also get the other two haplotypes that are from the other chromosome, so chromosome 10, that also had a contraction in one of the 10qa alleles. So it's really important to be able to distinguish, because remember, 4qa is the one you care about. That's the one that leads to DUX4 expression of the cytotoxic gene. So you have to know that you're looking at the right allele here that's contracted. You also would have a contraction here and would maybe miscall this if this wasn't assigned correctly to 10qa. And then we can also call the long alleles the healthy normal alleles in both of these. This is a look at the read length distribution for our ultra-long reads. So this is for all of the samples that we did, and then this is for one particular sample where we had the longest read. But we had a 566 kb read in one sample that was spanning the entire D4Z4 array. So that's basically up to 150 repeat units, which is about as long as these things can go. So you can get those from the ultra-long reads. But importantly, you don't need the ultra-long reads because the ligation sequencing, the assembled reads at 30 kb also almost match every single case that we tested. So I'm showing every sample, all of the allele calls, and then in yellow, this is from Coriell directly, their orthogonal testing and the repeat units that they have counted using BioNano and using Southern blotting. And then in green here and the purple row, this is our ULK repeat counts. So we think that that's actually more correct than the Coriell data, because these, again, are just direct measurements of the D4Z4 repeats. So we're using that as our ground truth. And then the assembled reads in the last column, it matches in every single case except for two. And in each one of those, it's off by one repeat and off by 2 repeats. So probably the assembler is collapsing on those 2 cases. But importantly, they're all correlated really highly with each other. So the ULK and the LSK is super highly correlated, as well as correlation to the Coriell data set. Okay, so lastly, we looked at the methylation and the chromatin regulation for these samples. So remember, in FSHD1, the repeat contraction loses methylation, and that's what leads to the hypomethylation and expression of DUX4. So we see this. So this is all 4 alleles. Now I'm plotting the methylation signal that you get from the base calling. You can see this oscillatory pattern. That's just the methylation at each repeat unit. So it's repetitive methylation pattern. But importantly, in the 4qa allele with the contracted D4Z4 repeats, we see lower methylation signal there, so hypomethylation, and that's consistent with the mechanism of action for FSHD1. If you zoom into these reads, these are now the raw reads. Red is the base called methylation, and then, so red is methylated and blue is unmethylated, so you can really clearly see that in the long repeats, we have hypermethylation, and then in the contracted repeats, we see hypomethylation and blue signal, so it's really clear that the shorter the repeat, the less methylation, and then the DUX4 expression is happening in these samples. I want to touch on one example where we had an FSHD2-like sample. So this doesn't have FSHD2, but it has a related syndrome called ICF syndrome. And what happens in FSHD2 or in ICF is you lose methylation signal across all of your alleles. So we see that. We saw low methylation in everything independent of the repeat length, and that's consistent with this mechanism. So we looked into the variant calls. We ran our epitome pipeline for human variation calling, and it comes up with 2 pathogenic variants in DNA methyltransferase 3b. These are known in the sample because, again, it has ICF syndrome. So it's good to see those, but that basically knocks out global methylation patterns. And because we have long reads, we can actually phase both haplotypes, so HAP1 and HAP2, and we see one pathogenic SNP or variant in the HAP1 and another one in HAP2. So this is a compound heterozygous mutation, which knocked out methylation and then leads to an FSHD2-like phenotype. Lastly, we did look at open chromatin or chromatin accessibility. So we have a protocol. It's on the website for looking at open chromatin accessibility. This is where you add exogenous 6-MA into your sample, and it basically adds 6MA exogenous marks into your open chromatin. And then so 6MA is not usually present in your genome, but when you do this, you should see 6MA increase in open chromatin regions. And we have base callers that are base calling models that are just part of our routine base calling. So we did this on the FSHD samples, and this is just one example, but we see now higher methylation signal, so this is 6MA signal now, is increased in the contracted allele, and that's consistent with that contracted allele having higher open chromatin and higher expression of DUX4, whereas in the longer allele, you see lower chromatin accessibility, more condensed chromatin. So again, that's consistent with the mechanisms that we would expect for FSHD1. Okay, that's all I have. In summary, basically, nanopore sequencing can give you haplotype-specific D4Z4 repeat lengths. It can give you the variant calls, the methylation that you need for FSHD samples. You can use the ultra-long kit and get really long reads to span the entire repeat array, but you don't need them. You can also just do this with ligation sequencing standard 30 kb reads that can then be assembled and put into the D4Z4 callers. Then we also looked at the methylation and chromatin stenciling to get more insight into what's happening at the chromatin regulation level for FSHD. So thanks to my team, the applications team, and everyone at Nanopore for this, and everyone for your attention. Happy to take any questions.